This is about the activation of the adaptive immune response in a draining lymph node, as you see here, that how the adaptive response is going to be developed. If you can see this picture clearly, there is a skin barrier at the top, and under the skin there are different cells. There is a dendritic cell, there is a macrophage, and different types of pathogens which are breaching that barrier of skin in the form of these different structure triangles. If you can see it, I would provide you that slide, then you can clearly see that. And these pathogens are first coming in contact with dendritic cells in the macrophages, and then these cells are migrating into this afferent lymphatic system. The afferent lymphatic system, or the vessels, are going to give way to these cells which are laden with these pathogens to enter into this particular structure, which is the lymph node here, right here. Okay. So these bacteria laden, these cells, the dendritic cells and the macrophages laden with these microbes enter into the lymph nodes along with these extra bacteria or their toxins or whatever products are produced in that tissue, in the connective tissue. All these enter into the lymphatic system or into the lymph node. And inside the lymph node, there are these cells, as I told you, the T cells and B cells. So this cell, the dendritic cell, is having some information about the pathogen, and now it is sharing this information with the B cells and the T cells, as you see here. And the B cells and T cells, when as there are millions of these cells right here in this lymph node, if this pathogen is recognized by a B cell receptor or a T cell receptor, then these cells are going to become effector cells in the form of plasma cells or in the form of memory cells. And these plasma cells are going to produce antibodies. These antibodies are going to be transported into the circulation, because this is an efferent lymphatic system which is going to take these antibodies into the circulation. So the antibodies have penetration into different tissues. These molecules can penetrate into different tissues, and wherever these antibodies would find this particular bacterium or this particular toxins which is there in the tissues, they are going to bind them, and neutralize them or do opsonization of the bacterial species or enhance that process of phagocytosis and killing, as I told you. So the adaptive immune responses are actually initiated in the draining lymph node. The B cells and T cells, effector B cells and T cells, are made inside the lymph nodes. Okay, and the B cells and T cells, you can, in other words, say that they functionally mature in these lymph nodes, and then these cells are going to, neutralize the pathogens throughout your body with the help of these antibodies, which are very specific in dealing with the pathogen. The spleen also is a very important, organ which is involved in adaptive immune responses, and it actually is dealing with blood infections. In most of the cases, for instance, in intestines or in other, secondary lymphoid tissues, the dendritic cells are sampling different antigens, and they are going to decide whether the immune system should respond against this particular antigen or not. They are going to train B cells and T cells in this art of immunity or providing protection, okay, by making them, turning them into effector T cells or B cells. But some of the pathogens, they can cross that barrier of innate immunity, and then some of the pathogens, for instance, can directly, rather than going through that infection through the skin or mucosal tissue and then being transported from connective tissue into the lymphatics, they can directly be injected into the blood. For instance, an insect can bite you, and the pathogen can directly be injected into your blood. What about the blood-borne infection? What would happen there? So nature has provided this particular structure for dealing with the blood-borne infections. We call it the graveyard of RBCs as well. This spleen, because it filters blood and it removes the dead or senescent RBCs, the red blood cells, from your system. Okay. But on the other hand, this is a very important secondary lymphoid organ, which is helping a great deal with adaptive immune responses. If you look at the structure of the spleen, there is a red pulp and there is a white pulp. You can compare this structure, this image with that one. The red pulp and a white pulp. So there are dispersed, patches of white pulp in that particular red pulp, as you see here. The red pulp is for management of the red blood cells, or RBCs. It means that the RBCs which are actually, dead, or they are not very functional, or they have got some problem, are going to be removed out of the system. When the blood enters through this arterial way into the spleen, they would be detected and removed. How it is going to happen? There are different mechanisms for that, because a cell, if it's going to become older, for instance, its membrane or its surface molecules can change in conformation, for instance. For instance, the phospholipid bilayers, normally it is, the fatty acid tails are inwards and the glycerol heads are outwards. But in cells that are going to die, or in damaged cells, this can be the other way around. The fatty acid tails at certain point of time, certain places can protrude outwards, which is a signal for different types of cells, including different macrophages, to sense such kind of structure and just take hold of them. This is a signal for a macrophage to just engulf this particular cell, because this has become non-functional, and just get rid of it. So this RBC management is taking place right here in the red pulp, while in the white pulp, adaptive immunity, or adaptive immune responses are taking place. And the process is just the same as it happens in the lymph node. The only difference between the spleen and the lymph node is that there is no separate lymphatic system in the spleen. These cells are going to enter through the arteries, and these cells are going to exit through the veins. There is blood supply, proper blood supply in the spleen, and this, inside the lymph node, if you compare with the lymph node, in the lymph nodes, the afferent lymphatics and the efferent lymphatics, that is a separate, system of vessels that are going to take the lymph inside, along with these lymphocytes, and to take these lymphocytes out of the lymph node. It has got no such structure. Now you can compare this structure or anatomy of the spleen, with the lymph node. So essentially, these are the same things. These different zones or centers which you see here, like the B cell area or the T cell area, it's the same as you see in the case of lymph nodes. Follicles are, follicles are structures where you can find some kind of cells, like the B cells, follicles, are areas in particular structures where you can find the B cells, or the T cells, follicles, are areas where you can find the T cells. The germinal centers are areas where these cells are going to turn into effector cells and propagate, So we call them the germinal centers. These structures are present in all the lymphoid tissues that we are talking about, the secondary lymphoid tissues, not the primary one. Some of the kids, they inherit this condition. They are asplenic. They don't have a spleen. Such kids are very prone to different kind of infections. This asplenic conditions happens because of some mutations in this ribosomal protein SA gene. In that, that mutation actually results in the loss of spleen in these kids. And if you don't have a spleen, these are the functions of the spleen. You will have problems with adaptive immune responses and removal of the red blood cells from the system. So it means that the individual would be immunodeficient, The individual would not be capable to deal with many different types of infections, for instance, Haemophilus influenza, for instance, or the capsulated bacteria would not be dealt by other systems in your body, They cause blood-borne infections, and for the blood-borne infections or the pathogens which are part of the blood now, if this structure is not present in the body, there is no other way to deal with it except if you do some kind of vaccination, So this is important for generation of the adaptive immune responses as well as the removal of the red blood cells from the system of this particular structure or the spleen is important. So some individuals can be asplenic, but some of the individuals can damage their spleen somehow, So in that case, usually splenectomy or removal of the spleen is recommended, because there can be a lot of blood loss, because a lot of blood is going into your spleen all the time. So if the spleen is damaged, it means that it's an open valve and you are going to die because of the blood loss, So that's why sometimes, the doctors would recommend to just, get that structure out of the bodies. We call it splenectomy or removal of the spleen from the body, So what do you think, would it have the same consequences as asplenic individuals would have? If you remove the spleen as a result of some accident or whatever, is it going to be the same thing, or it is going to make you prone to different kind of infections as it happens in the case of asplenic individuals or not? What is your intelligent guess? Have you heard about someone who has done or who has undergone this procedure, splenectomy? So what is your guess? Is it the same thing? Not having a spleen altogether and removing the spleen. How is it going to be different with respect to adaptive immunity?
So it's different because that person won't have some right to, but in that spleen and cloning, at least some part of their lifespan runs as properly. So they must be a little bit more immunosuppressed.
Yeah, that's the reason. She's correct, right. Many people, they are coming in contact with different pathogens, on daily basis. You know about that, okay? And I told you about this quality of the adaptive immune responses. that adaptive immune response is resulting into two things. One is antibody production, and the other one is keeping memory of the pathogen. Okay? So, if there is a memory of the pathogen in your body, you don't have to worry about reinfection with the same pathogen. Okay? So people who, adults for instance, if they undergo this procedure, splenectomy, they still would be protected from many different types of pathogens that they have come in contact with in their lifespan. Okay, they may have come in, contact with, for instance, different species of bacteria or different viruses, viral infections that he would have gone through. It means the memory of all those bacterial species and the viral species already would be present in his body as memory B cells. Okay? And the memory B cells are going to be activated once he would be reinfected with those viruses. Okay? If that virus is not different in nature, the same memory cells are going to respond. So he would be protected from a number of different infections, but a person who has undergone this ribosomal protein mutations, and he is born with this condition that he does not have any spleen in his body, would be prone to many different types of infections. Okay? The blood-borne pathogens especially, which are going to enter into your body through the blood. I'll give you some examples of these as well. So this is another structure which is a secondary lymphoid organ, and this is very important for development of the adaptive immune responses. The T cells and B cells which are found in these areas, the white area is for the B cells and T cells. The red area is for RBCs. So what are the white, what is the white pulp doing? The white pulp is a training ground for the B cells and the T cells. And the same kind of mechanisms are going to be initiated right here for making effector B cells or effector T cells is happened in the lymph nodes.